Right, good afternoon, everybody. I'm not sure how many of you were here before, so I don't need to do all the big long intro that we had before. But I'd just like to remind you, if you register for the book, there's a book that's going to be drawn after Dr. Mason's spoken called The Power of Us, which has an entry about Richard Fall in it. So if anybody'd like to be in to win that, it's that you register outside, and it's you'll be in the draw. There are 3 books to give away. They're good, substantial books too, so they're nice dipping in ones. Now our next speaker is New Zealand's leading brain stem cell researcher, Associate Professor Bronwen Connor, with her presentation Stem Cells and the Brain, Discovery, Myths and Reality. Stem cell research is a very exciting field that holds great promise for the future treatment of many diseases and injuries, including those of the brain. It is, however, a field that is surrounded by huge controversy, false promises and misinformation. Dr. Connor is a neuroscientist and an expert in stem cell research, and her presentation will demystify the issues on this hot topic. Dr. Connor will also discuss the truth behind clinical trials and the myths that surround stem cell treatment. In conclusion, she will update you about the exciting and groundbreaking research that has taken place in New Zealand, including her own recent breakthrough research that has made headlines around the world. And please join me in welcoming Dr. Connor to speak.
Thank you very much. It's a pleasure to be down here in Christchurch, a beautiful sunny day. So there we go. Okay, so what I'd like to tell you about today really is to give you an idea, hopefully when you leave here, what a stem cell actually is. And I want to talk to you about some of the research that we've been doing up at the University of Auckland. Our research focuses on adult stem cells, and so I'll talk to you about that research that we've been doing, but also, as Rosemary indicated, some of the really exciting stuff we've been doing in the last 2 years, which is known as reprogramming. Finally, what I'd like to do is just give you a bit of an overview and talk to you about what actually is going into clinic at the moment, and some of the myths that surround stem cell research and some of the products that are out there on the market at the moment. Okay, so what is a stem cell? Well, a stem cell is an unspecialised cell. Basically, it's the building block of all the cells and tissues and organs that go on to make the human body. It has the unique ability to grow into any type of tissue or body part and make up the mature human body. They're involved in development, and those are the embryonic stem cells, which I'll talk to you a bit more about in a minute. And they can also be found in many other types of adult tissues, where they seem to have a role in repair and regeneration of tissue. And these are the adult stem cells. So you can already see that there are 2 different populations of stem cells, embryonic stem cells and adult stem cells, and they are different in their capabilities. What I want to talk to you about today is the differences and the similarities between those 2 populations. So why are researchers excited about stem cells? Why has stem cell research become one of the really hot topics in neuroscience? Well, they have this really unique feature which makes them very exciting for regeneration therapy. And that is that they are capable of dividing and generating exact copies of themselves for a very long period of time. They can then be directed to turn into a specialised cell type. So we can take one embryonic stem cell or adult stem cell, we can put it in a dish, we can grow up millions and millions of exactly replica stem cells, and then we can give them chemicals and tell them to turn into a piece of heart tissue or a piece of brain tissue, depending on what we're interested in. Just to demonstrate this unique property, I've got a little video here. And I can find the mouse. There we go. So this is taken from Rusty Gage's lab at the Salk Institute. Oops, that's not what I wanted to do. Let's try that again. Okay, I'll push the right button this time. And here we have this, we're looking at the screen cell. These are adult stem cells, okay? These have come from adult brain tissue. And there's the stem cell dividing. It's made an exact copy of itself. A little time scale over here. You can see this sort of wriggling around. Split. There's another exact copy and another exact copy. So they're little clones. Going to do some more in a minute. There we go. They're really starting to get going now. So this is that exciting, unique feature that I was talking to you about. We just took one stem cell there, and we can grow up many, many more stem cells. And so what that means is that we can actually expand these up to vast quantities, whether they're embryonic stem cells or adult stem cells. Now, that video I showed you was an adult stem cell that was then dividing and multiplying. You saw it took about 48 hours, and we had probably about 15 or so cells there. Those are embryonic stem cells. That's magnified a whole lot faster. Okay, they're very, very fast at dividing. So the reason why we're so excited about stem cells is this unique property that they can self-renew, and we can make up large quantities of cells that we can then tell to turn into certain types of tissue. And obviously our interest is in trying to treat neurological disease and injury, so we can take stem cells and we can tell them to turn into brain cells, and not only that, into certain types of brain cells, which is a pretty cool thing to be able to do. Now this diagram here just shows you the different types of strategies, or two different types of strategies that can be used with stem cells. And traditionally, the main way that we think of stem cell therapy is the traditional transplant or grafting therapy, shown up here. And most people will think about embryonic stem cells. They've had a lot of attention in the media about using embryonic stem cells, the ethical issues associated with that, but using them for transplantation to replace lost brain cells or to repair spinal cord injury. But we can also get stem cells from adult or fetal tissue and from bone marrow and umbilical cord stem cells, and those are known as adult stem cells, and I'll talk to you about those in a minute. And these can be transplanted in, and they can make new replacement brain cells. The other thing I'll talk to you about is actually the fact that we have adult stem cells sitting in our brain. And these stem cells themselves, we hope to be able to actually utilise and promote the self-repair process to make new replacement brain cells instead of having to transplant in donor cells. The other thing that stem cells do, though, which is important, is that they can provide support and help cells and prevent them from dying. So as well as transplanting and replacing brain cells, particularly when you're using stem cells such as the bone marrow stem cells, they release chemicals that can help brain cells that are still there in the brain to become supported, to prevent them from dying, to help them grow new axons and connections, and also to prevent inflammation that often occurs in disease and injury. So they have a very supportive role as well. But therapy aside, stem cells also help tell us an awful lot about how the brain develops. So we can watch and look at adult stem cells and embryonic stem cells. We can understand and try and learn the different cues that are needed, the different chemicals that are needed to go from that really immature stem cell through to a fully developed brain cell. And that's very important for us to understand as researchers. So let me just give you an idea about the two different types of stem cells. First of all, we'll talk about embryonic stem cells. As I've said, these are the ones that most people will be familiar with from the popular media and the ones that have had most attention over the last decade. And you'll notice here I've got this important looking word, the word pluripotent. And this is very important. This is the main thing that's different between an embryonic stem cell and an adult stem cell. Pluripotent means the ability to turn into any type of cell in the body. Okay, that's what pluripotency means. So embryonic stem cells, because they are coming from a fertilised egg about 4 or 5 days after fertilisation, this is a diagram here. This is fertilised egg or blastocyte. Apart from the outer rim, all these cells in the middle are embryonic stem cells. That's how we all start life, okay, as a little bunch of embryonic stem cells. So these embryonic stem cells from the middle of the blastocyte can be taken, removed and put into a culture dish, and then they grow up, as we saw in that video, divide, continually expand. And then if we apply certain chemicals that we learn through developmental biology, as I said, we can turn them into cardiac muscle, into blood cells, and also into brain cells. The ability for them to turn into any of these types of cells, remember, comes from this word pluripotent. Okay, so that's an important difference. Now, as I said to you, the pluripotency, just to explain it a little bit more clearly, when we take stem cells and we're looking at them, using them for transplantation therapy, we don't transplant the stem cell. Okay, we transplant immature brain cells if we're talking about brain injury and brain disease. And this is important, and I'll explain to you why in a minute, but let me just walk through this process. So here we have our blastocyte, and we've collected our embryonic stem cells, and we've put them into a dish in the lab, and they're growing up. Now, we want to turn them into brain cells. So what we do is we take those embryonic stem cells, and as I said, we don't transplant embryonic stem cells. We take them and we apply chemicals, and we turn them into what we call a neuronal precursor cell. It's a very immature cell, okay? Basically what it is that what we're doing here is we're taking the embryonic stem cells and we're telling them what to become, okay? And we're saying to them, you are going to become a brain cell. We're going to give you all the chemicals and all the cues and all the factors that you need to become this immature brain cell. And then from the immature brain cell, or this precursor cell, they can go on and make mature brain cells and specific types of mature brain cells. We can take exactly the same population of stem cells. We can put in different chemicals. If we're interested in studying diabetes, we can turn them into pancreatic precursor cells. Because remember, they're pluripotent, okay? What decides what they become is at this point here, the factors, the cues, and the chemicals that they're exposed to. Now, it's very important, though, that we get the embryonic stem cells all turning into a precursor cell, okay? And this is the key point for probably what's holding up embryonic stem cells going into clinic. Embryonic stem cells by themselves, as I showed you in that video, like to divide, okay? But those video I showed you were adult stem cells. The embryonic stem cells divide really rapidly. So what we want to do here is we want to put the cues in and we want to tell them all to turn into neural precursor cells. Unfortunately, we haven't yet worked out how to do that with 100% efficiency. So if we've got, let's imagine the stroppy teenager embryonic stem cell who goes, nah, I'm not going to turn into a brain cell. I'm going to stay here and become a pluripotent embryonic stem cell. And we get a few of those rebellious stem cells that decide not to go down this path. If we transplant those cells, and a few of them sitting there are still embryonic stem cells, and they're still able to divide at rapid numbers, then the potential is there for those cells to turn into tumours. And that's the last thing we want to do when we're trying to treat people with a brain injury or brain disease or any disorder at all, is to put in a population of cells and then unfortunately say to them, I've cured you of Parkinson's disease, but you now have brain cancer. So until we can actually work out how to turn these cells 100% efficiently into precursor cells, that's probably the biggest challenge we still have to take embryonic stem cells into clinic. So the other type of stem cells which people are starting to become more interested in, which we've based our research on, are adult stem cells. So again, these are stem cells in the fact that they're uncommitted. They haven't decided what to become. But they're found in adult tissue or organs, and actually most of our organs in our body have stem cells. We have stem cells in the dermal layer of our skin, because our skin is constantly being shed and we're constantly making new skin. We have stem cells at the base root of our hair. We have stem cells in our liver, in our pancreas, and we now know that we have adult stem cells in our brain. So what these stem cells are there for is to help in a self-repair process. Cells die off in certain organs in the body, those stem cells go and they make new replacement cells, just a normal process of replacing cells. So they're involved in maintenance and repair. And this is really exciting because by discovering that we have adult stem cells in our brain and that they are trying to undergo a repair process, which I'll show you some studies that we've done a few years ago demonstrating this, we know that we could possibly take those stem cells and actually, or patients' own stem cells that are resident in their brain, and enhance that self-repair process. So instead of having to transplant in new cells, we might actually be able to get the brain to repair itself. But that's also a huge challenge. And I'll talk to you a bit more about that when I show you some of our recent work. So as I said, we've got stem cells, adult stem cells in all sorts of parts of our body. In our brain, we have what we call neural stem cells, or brain stem cells. And in the adult stem cells are actually divided up into these four groups. The neural stem cells, we have a very small population of what we call neural crest stem cells. They're an interesting bunch of cells. Those are the cells that are found at the bottom of your hair follicles and in the bottom layer of your skin. And also when your child loses their baby teeth, the dental pulp at the bottom of the tooth, if it comes out, that's full of stem cells. And it's sitting there to help promote the new adult tooth to come through. We also have a population of what we call mesenchymal stem cells, and these are the stem cells that are collected from bone marrow. So bone marrow basically is a huge source of stem cells. Mesenchymal stem cells can also be found in adipose tissue or fat tissue, breast tissue, and they go on to make bone, cartilage, fat, and connective tissue. And we have hemopoietic stem cells, or blood stem cells, and these have been known for a very long time in science. The blood system, of course, is constantly self-renewing, and these stem cells, hemopoietic stem cells, go and make new blood cells. So we have various different sources of stem cells. But the key thing about an adult stem cell is that it will only turn into a mature cell from the source from which it came from. Okay, so if it's a liver stem cell, it will only turn into liver tissue. If it's a brain stem cell, it will only turn into a brain cell. Okay, they don't have that pluripotency that I talked to you about with the embryonic stem cells. So we call adult stem cells multipotent. Okay, meaning that they can turn into all the different cell types of the organ from which they came from. Okay, so that's different to the embryonic stem cells. Okay, so just to go through here the similarities and differences between the embryonic and the adult stem cells. As I said, the embryonic stem cells, very exciting, they can turn into any cell type we want. That's that pluripotency. But the issue is that they can form tumours. The adult stem cells can only form into cells, mature cells from the organs that they came from. Because they already know that they need to turn into that type of cell, they don't form tumours. They're very unlikely to form tumours. They've never been reported in the literature yet to have formed tumours. Adult stem cells, though, unfortunately are more rare, often a lot more difficult to access, other than bone marrow and some of the blood stem cells. And they can be more difficult to grow in the lab. Okay, they're slower, like I showed you in that video. They don't divide as fast as the embryonic stem cells. They're adult, okay? They just take their time. They're not young, fresh embryonic stem cells. Embryonic stem cells grow easily, but as we're all aware, there are a lot of ethical issues and debate concerning the use of embryonic stem cells. Adult stem cells have the benefit that potentially you can get them from a patient, so that removes any issues of tissue rejection, the patient's getting back their own stem cells. Okay, so that's a big benefit compared to embryonic stem cells. Okay, so I've given you a little bit of an idea about what the different types of stem cells are. And what I want to do is just give you a little bit of a snippet of really what's actually about 13 years of research, which I'm going to put in a couple of slides. But hopefully it'll give you an idea about what we do and some of the exciting stuff that we've discovered. So we focus, as I said to you, on adult stem cells. And we're really interested in trying to find out, firstly, what those resident stem cells in your brain can actually do. Okay, why are they there? Why do we still have stem cells in our adult brain? What's their role? Just in the normal brain, what happens to them in everyday normal life? What can they do if you have a brain injury or brain disease? Okay, can they actually go and repair the brain? And that's the whole idea of this, what we call the self-repair process. And we've been working on that now for about 10 or 12 years, trying to enhance that self-repair process. I'll talk to you about some of our stem cell transplantation therapy work that we've also been doing, where we've compared the use of embryonic and adult stem cells. And finally, I forgot to put it on the slide, but I'll talk to you about some of our reprogramming work we've just done the last couple of years. Okay, so this picture here just shows you, so this is a rat brain. And the reason why I've got this slide up here is that this is probably the most studied brain in science. And unfortunately, for some of you out there, it is actually very similar to our own brain. So they're actually very smart rats. Okay. Here we have the two places in the brain. This is exactly the same. Even though this is a rat brain, this is exactly the same as in the human brain. There are two places where we have stem cells. Okay. And this little area up here, which is known as the subgranular zone, that's in the area of the hippocampus, which is where you learn and you make new memories. Okay. And I'm not going to talk much about those cells, but really interesting discovery of those stem cells there has led us to now understand that when you learn and you make a new memory, you actually make a new brain cell. Okay? And brain cells that exist make more connections onto other brain cells. So it's helped make the understanding of how learning and memory processes occur, it's given a lot of insight into that. The richest source of stem cells, though, is this area called the subventricular zone. Lines the fluid-filled cavities of the brain, and this is left over from development. Okay, so this is where, during development, the neural tube folds in. So these are left over. Well, they're not embryonic stem cells anymore, they're now adult stem cells, but that's residue of development. And this is the richest source. Now, they do something really interesting, and this isn't just in rodents, and I'll show you our evidence in humans, but these cells here, the ones from the subventricular zone, they go down this pathway up to the olfactory bulb or to the nose, which you might think, well, that's just really bizarre. And basically, this is just like a state highway, okay? They just zoom on down there. And you're thinking, okay, well, all right, why do we need to have stem cells going to our nose? Well, what happens in the nose is that about every 2 to 3 weeks, the olfactory neurons, or the olfactory brain cells in your nose, die. Because your nose, of course, is very exposed to external stimuli. So those brain cells don't survive for very long. So basically, in our normal brain, what these cells are doing is they're going and they're replacing brain cells in our nose on a constant basis. Now, you might be sitting there thinking to yourself, okay, I can understand why that happens in rats, because rats have incredibly good sense of smell, but as humans, we're pretty useless. But no, this pathway still exists, and I'll show you some evidence in a minute demonstrating that. So just to put this all into perspective, when I was a student at university, which I like to think was not all that long ago, we got taught that when your brain developed, when you reached about the age of 20, you stopped making new brain cells, and basically it was a slippery slope downhill, and you lost some humongous number of brain cells a minute, like 30,000 or something, which you don't want to think about. And that really that was it. It was a slippery slope downhill to old age, and if you were unfortunate enough to get a brain injury or brain disease, that was accelerated. But then in 1998, Rusty Gage and Piers Erickson at the Salk University, Salk Institute, made a discovery that demonstrated that actually that wasn't the case, and that actually the human brain could make new brain cells. Now, this had been known that mammalian brains were capable of making new brain cells. It had been known since the 60s. It had been shown in monkeys and cats and rats and pretty much every mammalian species that their brains could make new brain cells. But for some reason, being, the superior human race, we decided that our brains were so sophisticated and highly developed that we couldn't do that anymore. Well, Rusty Gage and Piers Erickson proved them wrong, in a very serendipitous experiment where there was a group of patients who were undergoing a new chemotherapy treatment for cancer of the throat. And they were being given a label which is known as BRDU. And what this label does is it labels cells that are dividing, okay? And they were using that label to track the tumour progression, okay? Because tumour cells divide. So the tumour cells would take the label up and the clinicians could see whether the tumours were growing or not. Rusty Gage and Piers Erickson thought about this, and they thought, hang on a minute, stem cells also divide. So if we were able to get post-mortem some of these patients' brains, we might be able to see whether there are dividing cells in the human brain. And unfortunately, a number of these patients did pass away, and Piers Erickson and Rusty Gage were able to get their brains. And this was actually 3 to 5 years after the patients had been given treatment. And lo and behold, what they discovered, and these are just going to look like black dots, but actually they're very significant black dots. These are cells, stem cells, that were labelled about 3 to 5 years ago that are constantly dividing in the adult human brain and in here in the subventricular zone. And they could also then see that these are making new brain cells. That was the first evidence that we ever had that our human brain was exactly the same as every other mammal's brain, and we could make new brain cells. And it really opened up a whole new field of thinking about the brain. No longer do we have this idea that it was a downhill slope of cell loss. We actually have continual cell development going on. The other thing, though, was that as I was talking to you about the olfactory bulb, and this, of course, was definitely well known in lower mammals, particularly rats. The other thing was that, okay, so we had stem cells in the human brain, but we didn't have this pathway to the nose because, as I said to you, our sense of smell is so terrible. You know, why would we have all these cells going up? So, okay, we've got stem cells sitting there, but really they're not really doing anything, not like in the rat brain where they're going up to make new cells in the nose. Well, at Auckland University, with Maurice Curtis and Richard Fall, we actually demonstrated that that wasn't the case. There is what we call a rostral migratory stream, or that motorway up to the nose, and it exists in humans, and here it is. Okay, so we are exactly the same as a rat brain. Sorry. So that's what they're doing normally. What happens if you have brain injury or brain disease? And a lot of groups internationally have been looking at this because this is pretty exciting if we can actually get the brain to repair itself. So at Auckland University, we did one of the first studies, or the first study in human brain tissue, to demonstrate that the human brain is using those stem cells to repair itself. And so at Auckland University, up at the Centre for Brain Research, we have the Neurological Foundation Human Brain Bank, which is a fantastic resource. We have hundreds and hundreds of specimens donated to us by very generous families and patients. And in particular, we have a very big Huntington's disease resource. And so this just shows you here that now this is a human brain. Okay, it's taken through here. This is a normal human brain. Huntington's disease affects the basal ganglia, this region here. And you can see in a Huntington's disease patient, this is very end-stage Huntington's, that whole area of the basal ganglia has been lost. It causes the chorea and the movement impairment in Huntington's disease. So we decided that we would have a look and see what happens in these Huntington's disease patients, looking at their post-mortem brain tissue. The reason we picked Huntington's disease is that we thought that of any disease, this was most likely to show a response to stem cells. The reason being that here we have that fluid-filled cavity I was talking to you about, and if I can hold my hand steady enough, the little white line there, that's the subventricular zone where all those stem cells live, right beside the area that is being degenerated in Huntington's disease. Those stem cells are sitting right there, okay? If they're going to go and do anything to help repair the brain, they are in a prime place for Huntington's disease. So along with Maurice Curtis and Richard Fall, we studied these post-mortem Huntington's brains and brains from normal patients. And again, the brown dots might not look very exciting, but they are very exciting to me. So here we have normal brain tissue, and here we have, this is that subventricular zone. In the human, we call it the subependymal layer. And you can't really see many brown dots because remember in the normal adult brain they turn over very slowly. And what we're staining for here, those brown dots are, is a label that shows a dividing cell. Okay, so you'd be pretty lucky to pick it up in a normal brain. But here we have different grades of Huntington's disease. You can see those dividing cells. Okay, they're dividing much more rapidly. We can start to see them showing up. And not only that, look how large this area's got. Okay, huge numbers of dividing cells. It's actually really expanded the area. So we have stem cells that are dividing because cells beside them are dying. And not only that, we went on and had a look and showed that they are going on to make new replacement brain cells. Pretty exciting. And that was the first evidence demonstrating that the adult human brain in a disease situation is going through and actually making new replacement brain cells. Okay, so I'm just going to walk through you now some of the studies that we did following this, just to try and understand this whole process. So we've demonstrated that this happens in the adult human brain, but obviously not to any extent where we have people recovering from a brain injury or where we have the brain repairing itself or while they've got Huntington's disease or Parkinson's disease. Otherwise, we just wouldn't have these neurological disorders. So obviously the brain is not able to do this sufficiently well. So we need to work out how we can enhance this process. And there are various steps along the way in this process. I'm just going to break down some of the areas that we've been studying. And as I said to you, this is about 10 years of research put into about 4 slides, which is a bit depressing, but anyway. So here we have our snapshot of the brain cut through. And this is just the normal brain, okay? So here we have those adult stem cells there that I was talking about, lining the fluid-filled space. And here we have the area in Huntington's disease that's lost. The cells are lost, okay? But here it's just a nice, normal, healthy brain. So the cells are just sitting there, they're just dividing nice and slowly. And we and other people have looked at Huntington's disease, stroke, Parkinson's disease, and various forms of brain injury. And this process is exactly the same regardless of what disease or injury you have, okay? So the process that the stem cells go through in response to brain injury and brain disease doesn't depend on what type of brain injury or brain disease you have. Simply the fact that brain cells have died. Okay, they have the same response. They don't care whether you've got Parkinson's disease, Huntington's disease, or you've had a stroke. They just respond to the cells dying. So what's the first thing they do? As I showed you with the human tissue with Huntington's disease, they divide and they make more of themselves. Okay, so here we now have an image. Just imagine, for example, we've had a stroke here and we've lost all these brain cells. The stem cells divide, make many more of themselves. They can do that really, really well. We don't need to help them do that. They're pretty good at doing that. The next 2 bits are the bits that are interesting, and that's where we focus on trying to enhance it. What they then do is they move into the area where the brain cells have been lost. Now this is interesting because normally those stem cells would be going down that motorway I showed you to the nose. Something's telling them not to go there and telling them to go into there. And so we did a bunch of research. A very talented PhD student in my lab, Renee Gordon, worked for 4 or 5 years on this question. What tells the stem cell to change direction and go somewhere else? And if we can understand what it is, can we make more of them change direction and go in there? Because they don't all go in there, okay? We still have some of them going up to the nose, actually most of them going up to the nose. So here's my little diagram that I showed you before. Remember, we've got the stem cells in the subventricular zone that we're looking at here, and they normally go up that pathway to the nose, up that motorway. And using some nice fancy technology where we can actually stain cells green and watch them move, you can see this is just a normal brain. Here we have flipped around, they're going this way. Here's the nose. You can see the cells. These are individual cells going along that pathway. Okay, this is just caught in a snapshot of time, and they're just travelling along. This is what they normally do. Okay, so in your brain, normally you just have cells trickling along like that. Normally they're not green though. if, for example, you have Huntington's disease, look what happens. That pathway just looks like Auckland's motorway system on a Friday night. It's just absolutely chock-a-block, all going up here. So remember we had all those stem cells dividing, so that's a huge number of stem cells dividing, but they're all going to the nose, which is like, hang on, you don't want to go to the nose, that's not where you want to go. So we've got all these cells going up here, but we also have, so this is, once again, here's where those cells reside. They're going to go up along here to the nose, but here we have them actually moving into the area where the cells have died. So we have a population, probably about 25% of them, that decide not to go on the motorway, and they go out into the area where the cells are divided to make new brain cells. Obviously, though, we want to try and get rid of this traffic congestion, take some of these guys and put more of them into here. Okay? Get them off the motorway and put them into the suburbs. So that was Renee's big question. And they do this like little magnets. They know exactly where to go. Very, very clever. How do they know where to go? Well, this little video here I'm just going to show you, and this is real-time imaging. Okay, so these are cells really moving. This will make you think about your brain a bit differently. This is actually going on in your head right at the moment. And this is what it looks like. There they are, pottering along, okay? So you thought your brain was just sitting there quietly. I'll show you. I'll click it once more time for those of you that didn't quite catch it. Here we go. Okay, that's what they normally do. I think they're pretty cool. I love these little guys. They just zip around. So we need to try and get them to zip directly to the area where the brain cells are dying. Okay, so how do they know where to go? Well, this is one slide that summarises someone's entire thesis. Basically, she'll love that, I know. Basically, they know where to go because of inflammation. Okay, inflammation is not always bad. Inflammation is there for a reason. One of the reasons it's there is to tell stem cells where to go. So if you have brain injury or brain disease, one of the natural things that happen is that you have an inflammatory response. Okay, just like you have an infection on your hand, well, that happens in your brain as well. And what gets produced are chemicals, very special chemicals that we call chemokines. These chemicals, their job is to tell immune cells to come to the area to mop up the damage, to clean up. So it's the clean-up crew. So the first ones that come on and they send out these chemicals and they tell the rest of the clean-up crew to come in. They also tell the stem cells to come in. The stem cells can respond to a huge number, so there's about 20 different chemokines, depending on what type of injury you've had, what type of inflammation. They can respond to all of them. So that's why you have some of them going that way, mainly because they just haven't got the signal, they didn't get the message, and the others go, oh, we need to go there, because that's what the chemokines are telling us. There's a high level of chemokine expression, we've got to go down there. Eventually, this inflammation dies off if you've had a stroke, for example, and they stop coming in. So there's two ways that we could approach this. We could keep these chemokines going for longer. Problem is, though, that we can also make the inflammation get worse by doing that. Or we could somehow try and make these cells more responsive, more of these cells responsive to these chemokines. And those are different things that we're trying to do at the moment. The other thing, the thing that then happens is that we get these new brain cells developing. So the cells have moved in, they know where to go, and then they start to make new brain cells. And here's a picture of a newly formed brain cell. It's green because we've labelled it with the dye in a rat to show that it used to be a stem cell and now it's a mature brain cell. So it's pretty cool, but unfortunately the sad news is that less than 1% survive for more than 2 months. So the brain is trying to repair itself, it's doing a semi-decent job, it gets there, the cells that do get there do make new brain cells, but eventually they die. And the big question is, why do they die? Well, once again, it goes back to inflammation, unfortunately. Inflammation goes on, the clean-up crew comes in, they mop up the dead cells, the debris, they get a bit carried away, and it turns into chronic inflammation. And chronic inflammation will kill nice, healthy cells as well. These new brain cells are very susceptible, so the same process that told them to come in then actually kills them. So what we need to do, obviously, and we're working on different techniques to try and do this, is not to completely turn the immune system off or the inflammatory system off, because we need it. We need it to tell the brain cells where to go. But we need to try and stop it from becoming chronic and killing all these brain cells off. Let's help the brain actually do its repair job and not have the, come here, I will kill you. So one way we can also do it, though, is we can play around with the immune system, but we can also try and provide chemicals or factors that help cells survive, make them stronger. And these are chemicals that happen during development, and one of them in particular for the area in the basal ganglia is a chemical or a factor known as brain-derived neurotrophic factor, or BDNF. And we thought actually during development, BDNF is needed to make new GABA cells in the striatum in patients with Huntington's disease. BDNF is required to make new GABA cells in the brain, and those are the cells that are lost during Huntington's disease. So we thought, what happens if we put BDNF in? Can it help protect those newborn brain cells? And the answer is yes, it does. If we can get there. Hello. Oops. You seem to be stuck. Okay, well, if I could go to the next slide, I would be able to show you that when we put BDNF in, what we actually get is a whole lot more stem cells being made and a whole lot more new brain cells surviving. It's going to be difficult to continue the talk without the pictures. Hi.
It's just crashed on your head.
Yep.
This is great.
Oh, now it's completely dead. Yep, it's going there. Okay.
Okay, turn the shuttle back off. Open the shuttle again.
Oh, there we go. Okay. So, oops. Okay. So, we showed that we could make a whole lot more stem cells, and a whole lot more of them survived and made new brain cells, and those brain cells survived for a lot longer. So by delivering BDNF, or brain-derived neurotrophic factor, we could actually help promote that self-repair process. Now that's something that we're looking at making more accessible for clinical therapy. Finally, I just want to show you some of our transplantation therapy stuff that we've been doing, and we were very interested in, so this is the traditional way of thinking about stem cell therapy, obviously, is to transplant them and make replacement brain cells. We were interested, though, as to whether embryonic stem cells or adult stem cells would be better, because there's a big debate as to which to be used. So we did a study where we compared both types of stem cells, adult and embryonic, and transplanted them into a Huntington's disease rat. So these are the embryonic stem cells. Now remember I told you that what we do is we take the embryonic stem cells and we turn them into these immature brain cells or neural precursor cells, and those are the things that we actually transplant. And that work was actually done at the Howard Florey Institute at the University of Melbourne by collaborators of ours there, Mirella Dettori and Professor Mel Horn. They have human embryonic stem cells readily available and growing in their labs. We transplanted them into the animals, and we've stained them green so that we can see them nice and easily. Of course, stem cells are not normally green. But this is a transplant, okay, and it just completely fills the whole area where the cells have been lost. Pretty impressive stuff. And they make vast numbers of new brain cells, and specifically those brain cells that have been lost in Huntington's disease, the GABA cells. Very, very impressive. But, there's always a but. I talked to you about the issue of the pluripotency and the tumour formation. We had about 10% of our animals, this is a tumour, glioblastoma, about 10% of the animals formed a glioblastoma. That is using the technology from the Howard Florey Institute, which is one of the best in the world at trying to purify stem cells into brain precursor cells. Okay, so this is still the issue here. These cells have huge potential. We've got to get over this, around this issue. In saying that, our adult stem cells are pretty impressive as well. We transplanted them in, and these are probably not going to mean an awful lot to you, so I'll walk through it. Here we have our transplanted our adult stem cells, and they turn into two different types. We've got astrocytes in our brain, which is support cells. They needed to be there. They turn into support cells. They also turn into new brain cells or new neurons, and specifically, again, the same types of brain cells that are lost in Huntington's disease. What was really exciting for us, though, is there's no point transplanting new cells in if the patient's not going to see any clinical improvement. And we can do tests with rats. Rats are very good. They like to run through mazes and climb cylinders and do all sorts of things, particularly if you give them Cocoa Pops. They love Cocoa Pops. And they don't like cheese, by the way. They love Cocoa Pops and peanut butter. This one here just shows basically the pale bars are animals that we didn't treat, so they're very impaired. And within 6 to 7 weeks after transplanting, you can see that the transplanted animals, significant improvement in their motor function. Okay, they're starting to regain their motor function back. And this is probably best shown here with this study. These are the animals that didn't receive treatment. Their motor function is progressively getting worse. We transplant the animals and actually they go back to literally normal motor function. Okay, very impressive. Okay, so I'm just going to finish off telling you about a bit of science fiction, which is actually stuff that we're really excited about and that we're working on now. And that is the field of reprogramming. Okay, so might be the first time you've heard of reprogramming. You heard it first right here. It's pretty exciting new technology, and was actually invented, I suppose. Invented by Yamanaka from the University of Kyoto. He actually won the Nobel Prize last year for this. Okay, what Yamanaka did back in 2007 is he took skin cells, so basically just a skin biopsy from people. He put in some genes, and these are genes that are found in embryonic stem cells. These are genes that tell a cell that it's an embryonic stem cell. Put them into the skin cells, and he produced cells that are identical to embryonic stem cells. Okay, so he took skin and he turned it into embryonic stem cells. That's pretty incredible. We can then take those, we call them induced pluripotent stem cells, IPS cells. You can then take them just like you take embryonic stem cells, this is the same pathway, give them the chemicals, turn them into neural precursor cells, and they turn into mature brain cells. Okay? So we don't need embryonic stem cells anymore, we just need a piece of skin. That's pretty cool. But we were pretty excited by this stuff, and we were sort of sitting down in the lab one day and talking about it and thinking, well, what can we do? We should start doing this stuff in the lab. But the issue, again, having had our experience with embryonic stem cells and the tumour formation in our rats, we thought, well, we don't want this stage. We don't want to go all the way back to embryonic stem cells and then go back to a brain cell. We actually want to just go right that way, like that. And so that's what we did. And so what we developed about 2 years ago was a technology where we put in just two genes. There's two genes here. They're found as an embryonic stem cell turns into an immature brain cell. We can take skin from adults, we can put these two genes in, and we can make neural precursor cells, or what we call induced neural precursor cells. And then we can expose them to chemicals and we can turn them into mature brain cells. We skip that pluripotency, we skip the risk of tumour formation. Okay, we just go straight to what we actually want to repair neural injury and brain disease. So just to show you some pictures here, these are skin cells. This is what they look like. Okay, so take a skin biopsy and you put it into a, sort of turn it up a bit, and you put it into a dish in the lab, and these are skin cells. We put our 2 genes, SOX2 and PAX6, and they turn into these little balls of cells. Now that's not a big giant cell, that's actually, remember I showed you the cells dividing right at the beginning of that video? What they then like to do is they form balls. Okay, they like to sort of hug together. So these came from one cell that is then divided many, many times. And here we go, it's our ball of many hundreds of cells, our induced neural precursor cells, and then we can turn them into, and these are mature brain cells here. And this in particular is a GABA cell that we've made, and they show the same electrical properties as a mature brain cell. So they look, act, and sound and feel exactly like a brain cell. What's really exciting for us is that we're the only group in the world that has done this with adult human tissue, and we're the only group in the world that's only used 2 genes. The rest of the world internationally are still working on rat cells, and they're still using up to 5 to 7 genes to be able to do this. So this is a bit of New Zealand science. It's pretty exciting. What we can also do is we can take patients' own cells, and we've just done this recently with a Parkinson's patient, skin biopsy. We can take skin biopsies from patients such as Parkinson's disease, and this is a dopamine cell that we've made. So the dopamine cells are the cells that are selectively lost in Parkinson's disease. We've made new dopamine cells from that Parkinson's patient's skin biopsy. Really exciting stuff that we can now go and do. We could transplant those cells back into that patient because it's the patient's own cells that have come from them. The other thing we can do, which we're pretty excited about, is we can actually model the disease. And what we mean by this is we can take these cells and we can start to look at them and see how they're different to normal cells. And what's exciting is we can't do this, we can't get human brain cells and neurons in a dish, they don't survive. We can do this though, we can grow these up. These guys do survive in a dish, and we can start to look and see how they're different from normal cells and hopefully develop new drugs. Okay, just very quickly at the end, I just want to touch on some of the myths and realities about the clinical progression of stem cells. And I just put this cartoon in here. Nothing that a few stem cells and 75 years of research can't fix. Well, I'm hoping it's not going to be 75 years of research. But, you know, there's huge potential here. We just don't know how fast this field's going to go. I think reprogramming is now going to really be the way to go. Removes ethics, and it's got huge potential. Okay, so let's talk about current clinical trials. What's currently available with stem cells? Okay, so a clinical trial, just for those of you who are not familiar with what a clinical trial actually is. Clinical trial is a proper procedure that's used to establish whether a new treatment is safe and whether it's effective. You enter into a clinical trial with 3 major questions. What are the unwanted side effects, or what are the side effects? What is the risk level of this therapy? And what is the beneficial level? Basically, what you're looking at is the risk-benefit ratio. And if you have a current therapy on the market, so for example, we're thinking of a new pain relief drug, if you've got a current therapy on the market, it has to have less risk and more benefit than that current therapy. With neurological diseases and brain injury, though, of course, we really have no real treatments. So the risk-benefit ratio we're talking about here is there risk to the patient's health? Okay, not converse to a current therapy, because there really isn't a current therapy to judge it to. Clinical trials are based on extensive preclinical research, so years and years of people like myself doing stuff in the lab over and over again, other groups repeating our studies, demonstrating that everyone can do the same thing. And it goes through rigorous peer review. What I mean by this is that we have our own colleagues look at our work before it gets published and criticise it, and sometimes very harshly criticise it, and make sure that actually it is what it says it is. Okay? So we have to actually walk the walk and talk the talk. Now, stem cell therapy is not a drug, is it? It's a very different way of thinking about treatment. It's not a drug or a medicine, but it's still a therapy. Because of that, it must still obtain American Food and Drug Administration, or FDA, approval, or the equivalent body in European legislation. Okay? Because what we're thinking about here is still comes under the medicine's clinical therapeutic procedures. Okay, so must have FDA approval, which means it goes through sometimes a very, very long, rigorous assessment of all of the preclinical research that's required, and sometimes they have to go back and do more to prove that it's ready to go into clinic. So what is actually available? Well, bone marrow, and I've just put umbilical cord stem cells here, but really I want to talk about bone marrow, and I can answer any questions about umbilical cord stem cells if anyone has any. But bone marrow has been used now for a very long time for the treatment of leukemias and rare blood disorders. We know what bone marrow does. We know that it's effective and that it's very safe. And because of that, it has skipped a lot of the FDA procedures, and people are now able to start to use it for clinical trials because they're stem cells, okay? And they're being put into clinical trials for a huge range of disorders I've listed up there, including some in the brain or the central nervous system, multiple sclerosis, motor neuron disease, stroke, and cerebral palsy. Bone marrow stem cells are very exciting because they're autologous, meaning they come from the patient. They're easily accessed by a bone marrow biopsy, and as I said, they've got current approval. The thing here, though, is remember right at the beginning I said to you that stem cells can be used either to replace neurons or to help support existing brain cells by producing chemicals and reducing inflammation. Bone marrow stem cells, there's very, very little to no evidence that they turn into brain cells. Okay? So when you have a bone marrow transplant for a neurological disease or injury, it's not going to make new brain cells, but it's going to produce chemicals to help support the existing brain cells, help promote their survival and to reduce inflammation. Now what I've listed up here is the 4 different clinical trials that have been approved using brain stem cells, and you can actually go and look at these yourself. You just type in clinical trials FDA and you get this huge, great big website, which you can then break down into diseases or cell types or stuff like that. So it's readily accessible for the general public. 3 companies here I've put up the top. These are not embryonic stem cells that they're using. These are adult stem cells that have been taken from fetal brain tissue. Okay, they're considered adult brain cells. Been used, they're all in phase 1 trial at the moment, so that's just general safety. Okay, are they safe? Not whether they have any benefit, not whether the patient gets better, just are they safe? And they've been looked at for a range of different neurological disorders. The one that everyone was very excited about was from a company called Neurons, and this is the only embryonic stem cell clinical trial to have actually got FDA approval. They make a cell type known as oligodendrocytes, which makes new the coating on the axons in brain cells, and they're useful very effective for treating spinal cord injury. They were able to make oligo precursor cells, and they were looking at putting them into spinal cord injury. Originally in beginning of 2012, they got FDA approval. Sorry, 2011 they got FDA approval. Then the FDA turned around and said, hang on, we're going to hold that until you can show us that you can safely remove the cells if they start to form tumours. They went away, they did some stuff. We don't know what they did. It's all confidential. They got their approval put back on in August. By December, they had closed down the whole trial. Okay, it's been halted, apparently for financial reasons. So there is currently no clinical trial for embryonic stem cells underway. But if you Google stem cell therapy, you will come up with a large list of clinics that claim that they can treat huge ranges of diseases, usually lists and lists, everything from asthma to Parkinson's disease to fatigue with stem cells. And you have to be very cautious about these clinics. So they claim that they can treat multiple diseases with the same cells. I will tell you right now that an embryonic stem cell and different types of adult stem cells will not be used to treat everything. Okay? Bone marrow stem cells might be used to treat one thing, embryonic stem cells might be used to treat another thing, a different type of adult stem cells might be used to treat something else. Okay, there's little evidence that one source of stem cell is going to do everything. So if you see that, you might want to be a little bit cautious. These clinics also base claims on patient testimonial. Now, I said to you before, clinical trials are based on years and years of preclinical research, and usually phase 1 to phase 2 clinical trial. Safety is phase 1. Phase 2 is, does it seem to have some effect in quite severely affected people? Patient testimonial is not a reliable source, unfortunately. There's a very incredible thing known as the placebo effect. Okay, people want to get better. Okay, and they sometimes believe that they are getting better. A clinical trial is done properly, where you go through a university or a clinical trial centre, you will be tested before you have a clinical trial. You will be tested routinely, monthly or 6 monthly or yearly after your procedure. All documented, all blind, so nobody knows who's had what, and all completely open for peer review and scientific analysis. What happens with these clinics quite often is that the patients go to the clinic, no pre-testing, they have stem cells given to them, and they go home, and nobody ever sees them again. Okay, that's not a way to work out whether you have an effective therapy. Quite often the source of cells or how the treatment is done is not clearly documented, and there can be some question as to whether you're even receiving stem cells. And generally there's claim that there's no risk. Well, I'd like to say that there is no risk, but we've already talked about embryonic stem cells producing tumours. Adult stem cells, there hasn't seemed to be any risk so far reported, but this is still early days. We haven't put many stem cells into people to actually know what those risks are. And that risk profile has to be done in a very professional, very accurate manner through a proper clinical trial. Usually these clinics ask for payment of the treatment, or there are hidden costs. Now, if you go into a clinical trial, you're a volunteer. You don't pay to be part of a clinical trial because you're a volunteer. They want you, okay? And you should try and decide whether you think you should be paying for something that's not proven, okay? And these can be many thousands of dollars. The other things I always remind people to think about is that if you go overseas to have stem cell therapy done in a clinic and you come back to New Zealand and you have a bad reaction, your medical insurance will not pay for you, okay? So you need to be cautious about these decisions. There's usually no follow-up procedures. As I said, the patients go over there, a couple of days later they're sent back home. If we don't report, if we don't follow up the patients, maybe these clinics have found the holy grail to treating Parkinson's disease or Huntington's disease. But if they don't follow it up properly, if they don't report it properly, no one else will know. No one else can also check it. No other clinic can go and say, okay, well, let's try this as well. Can we do it also? That's not how science and clinical therapy progresses. The other thing I just want to touch on very briefly, because these are available in New Zealand and you need to be aware of them, is these stem cell pills. So one of them, Colostem, has this statement, which I wish was true, stimulating the release of adult stem cells from bone marrow into the circulation to slow aging process and repair in areas of the body suffering from degeneration and injury. Sounds great. So Colostem is a colostrum product. Colostrum, of course, is produced by the mother's milk, breast milk within the first couple of days after birth. It's required to stimulate the immune system. Very beneficial during that period of time, has very little effect later on in your body, and it has no effect on bone marrow and stem cell release, and that's been very clearly shown. The other one, which is ever so slightly more dangerous, is Stem Enhance. It's a blend of two compounds from a aquatic weed, which name I'm not going to bother trying to pronounce, AFA. There's no scientific evidence that AFA affects bone marrow stem cell release or movement through the body. And actually there's been a couple of incidences which has demonstrated that AFA, and if any one of you is familiar with this, is red algal bloom, okay, which is known to be highly toxic. If you take large amounts of it for a long period of time, it can cause liver toxicity and kill you. Now, these are food supplements. They don't need FDA approval. They can come into New Zealand under the natural medicines, okay? So there's no restriction or requirement on them to be tested, but be careful, okay? The colostrum probably is not going to do anything to you. Stem enhance may be a little bit more dangerous. Okay, so just to finish up, if any of you have any questions, there is the International Society of Stem Cell Research, international body of science of stem cell researchers, so concerned about some of these clinics and people going overseas and possibly having unnecessary treatment, that there is a website you can go to, I'll put it up in a minute, facts about stem cell treatment, and also if you really want to have an insight into one of the clinics, you're really interested in whether or not it's actually real, you can contact ISSCR, and a professional stem cell researcher will actually go and talk to that, will approach that clinic on your behalf and ask all your questions and give you proper feedback. Okay, so don't feel that you're alone. There are people out here who will help you make these decisions or give you advice in regards to whether it's beneficial or not. So just in conclusion, stem cell therapy is very exciting and it is a real possibility. But we've got a lot to learn about stem cells. Okay, we still don't know an awful lot about them, and we definitely don't know much about what happens when we put them into the human body yet. We need to be sure that the long-term safety and effectiveness of stem cell therapies before we put it in the clinic. And a clinical trial is the only way of doing this. The thing I always say, and I just want to remind you, is that we need to do lots of good research. We need to do proper clinical trials. One thing to think about stem cell therapy is if you take aspirin and you have a bad side effect from aspirin, makes you feel sick, you just stop taking aspirin and that goes away. If you have stem cell therapy, we inject stem cells into you, and you have an adverse reaction or adverse event, we can't take those stem cells back out. Okay? We need to know it's going to be safe. The clinics may have found something, but they're not sharing it and they're not doing the proper type of science, so they're not helping with the development, and many of them offer empty promises. And unfortunately, because it might do me out of a job, but it would be wonderful to see it, there is no over-the-counter pill, stem cell pill available yet. Maybe that's what we want to aim for in the future, but it's not here yet. Okay, thanks. This is all my team over the years. Richard Fall, Director of the Centre for Brain Research, collaborators in Australia and America. Neurological Foundation have been very, very supportive of this research, and just the supporting information I was saying to you. Stem cells, just some basic information. This is the ISSCR patient guide. Clinical trials, if you want to know, just go to this website and have a look. But also, please feel free to contact the Centre for Brain Research. Got any questions, you want to get any advice, we're always more than happy to provide any information we can. Thank you.
Thank you. That was absolutely fascinating. I really felt I heard a lot of things I hadn't heard before, so thank you, Bronwen. That was wonderful. Now, I'm sorry we've gone over time again, so we won't have time for any questions. And I think people have got, you were sort of organised to do something out in the green room. But if, maybe when Bronwen's finished, she might be able to spend a few minutes outside and answer some questions. But you've got all these places that you can find information out. So thank you for listening, and thank you again, Bronwen. That was really interesting. So thank you. Sorry about the time. I'm quite happy not to have questions. I find it a bit difficult.